Okay, so for some reason, the camera is not working now for this part, but we kind of need to get this out. So, um, picking up where we left off, the plasma membrane lets certain things in or out of the cell. And so, how does it do this? The plasma membrane has what's called selective permeability. Only some stuff can cross, or some stuff crosses more easily than others. So it's permeable, but it's selectively permeable. Only some substances can cross, and some cross more easily than others. So if you were to watch one of the Crash Course Biology videos, the example that he gives is that the cell is like a club. And not everybody could get into the club. You know, there's some restrictions. You have to be over 21, whatnot. You can't certify or else you get kicked out. So I think that is a great metaphor. If the club is like the cell, only certain things can get in and out. The membrane would kind of like be the walls. And then there are different proteins that can let things in or out if they can't get in or out on their own. And so that would kind of be like the bouncer, or if they know somebody. I don't know. This metaphor only goes so far, but it's selectively permeable. Not just anything can get into the club. So molecules that are hydrophobic, and they're hydrophobic because they're nonpolar. Again, remember, the importance of water. It keeps coming back, right? So water is a polar molecule. If another molecule is nonpolar, that's going to lead it to not really associate with water. So hydrophobic or nonpolar molecules are able to dissolve in the lipid bilayer and pass through the membrane on their own, and they do that fast. And so that's because they are hydrophobic and nonpolar. They are able to cross that middle layer of the phospholipid bilayer. And that middle layer is hydrophobic. So that's kind of like the biggest barrier, is whether or not a molecule is hydrophobic also, and then it could go in on its own. So for example, molecules that are hydrocarbons, so hopefully you remember that as well, longer chains of carbons and hydrogens, they are nonpolar, and so they're hydrophobic. They're easy to pass through the membrane. Other molecules that are nonpolar, like carbon dioxide and oxygen, can pass easily through the membrane. Molecules that are polar and hydrophilic, they don't cross easily because of that middle hydrophobic layer in the middle of the membrane. So it's kind of like, it mixes well with water, but it just can't get through. It bounces back from that hydrophobic interior of the membrane. So stuff that is polar, like sugars, water, and ions, pass through very slowly, if at all. So whether or not something can get into the membrane on its own depends on the size of the molecules, how small they are, and then also the polarity, if they are nonpolar versus polar, because that means it's hydrophobic versus hydrophilic. Hydrophilic substances can cross more quickly if they pass through a transport protein. So that's kind of like if there was another, like a door to the club. I don't know, this metaphor falls apart. So hydrophilic substances can cross membranes more quickly by passing through transport proteins. And there are two general categories of transport proteins. Channel proteins have a hydrophilic channel, kind of like a tunnel. So channel, think of tunnel. Channel proteins have a hydrophilic channel that certain molecules or ions can use as a tunnel. Carrier proteins, these are proteins that they bind to the molecules and then they change shape a little so that it can allow them to cross the membrane. So here, for example, this is a channel protein, so like a tunnel. Molecules just really go through the tunnel. In this figure, this is a carrier protein. So this protein has a shape change to carry this molecule in, changes shape, and then spits it out on the other side. I thought I had a picture right here. So yeah, channel proteins have a tunnel in the middle. Carrier proteins have to change shape to shuttle them across. There are channel proteins that are called aquaporins that greatly increase the rate of passage of water molecules. So again, water is polar, so it's going to kind of have a harder time. It's not going to go as fast through the membrane. So these channel proteins, aquaporins, help water pass through the membrane faster. They are made of 4 polypeptide subunits. So remember, proteins, they have the 4 levels of structure, and so a polypeptide is, a shorter chain of amino acids, and then the full functional protein usually has multiple polypeptide parts that come together. So aquaporins are made of 4 polypeptide subunits that each form a channel for the passage of water. And overall, up to 3 billion water molecules pass through per second. And the transport proteins move only specific substances. So for example, glucose only transports glucose, not anything even similar. So they won't transport fructose, which is actually an isomer of glucose, meaning they have like the same basic components, but its shape is a little bit different. It's like remember what an isomer is? It's like a mirror image. So glucose carrier proteins will only transport glucose, not fructose, even if it looks similar. Aquaporins will only let water through, not H2O2, hydrogen peroxide. So each protein is in charge of just the one thing it's in charge of letting in. So selective permeability of a membrane depends on both the lipid bilayer and what specific transport proteins it has. So again, coming back to this idea of how some things get in or out of the cell, and these are smaller molecules. So some ways don't require energy to get in or out of the cell, and so we call that passive transport. And some ways to get in or out of the cell do require energy, so we call that active transport. So here in the figure, you can see examples of, on the left, passive transport. There are 3 examples. And then on the right, you see an example of active transport, and that molecule of ATP that the cell uses as energy is used. So I'm going to break down each one of these things. So Roman numeral 1, passive transport. We're going to talk about these 3 kinds of passive transport and another one. And then Roman numeral 2 will be active transport. So hopefully this helps organize your notes and how the concepts fit together. So passive transport is the diffusion of a substance across a membrane with no energy investment. And the rate of diffusion depends on the concentration gradient of that molecule and the permeability to the specific substance. So first of all, is there a protein or can this molecule pass through on its own? If it can't pass through on its own and there's a protein, what kind of protein is it? So the rate of diffusion depends on if it's permeable, and then also what's called the concentration gradient. And so we'll talk about what that is. So the first case of passive transport is diffusion. Diffusion is the movement of particles of any substance so that they spread out evenly into the available space. So here is an image. So here are different molecules of orange dye. And if they start out on the left side, they're going to diffuse and kind of just spread out. They randomly bump into each other. They don't like intentionally move a certain direction. They just bump into each other and then happen to end up going the same direction. So diffusion is really just the molecules bumping around and then spreading out so that eventually they're equally spread out. And so here on the right, you see the molecules equally spread out. So diffusion is the movement of particles of any substance so that they spread out evenly into the available space. Say, for example, you have an aroma diffuser or an oil diffuser. I think it's called an aroma diffuser. And so you have like a little concentrated scent. And then what that machine does is it warms it up. And then so the oil or scent spreads out throughout the room. So at first, you're only really going to smell this scent as you're closer to the diffuser. But over time, it spreads out throughout the room. And then if somebody just walked into the room, they would be able to tell, oh, that smells good. Do you have the lavender thing? So that's literally the principle of diffusion is like the molecules will start one place and then they'll randomly move and bump into each other and spread out until they're evenly spread in the available space. So these molecules move randomly. They just happen to bump into each other, and then so they end up spreading out. So although each molecule moves randomly, diffusion will end up kind of being directional. And at what's called equilibrium, many molecules, it'll be spread out over the entire space equally. And things will still move. It's not like things are going to stop moving. That's why it's called dynamic equilibrium. It's that things will keep moving, but they will stay equally spread out. So at dynamic equilibrium, as many molecules cross the membrane in one direction as in the other, because we're talking about cells. So it's going from either inside to outside the cell or outside of the cell to inside the cell. So there's going to be a membrane that is separating everything. So that's why in this picture there was these little gray lines crossing, and so this represents like a membrane separating this area of water from this area of water. And so if this membrane is permeable to this kind of dye, so if it's big enough for these to pass through, then they will. And so the molecules of dye will bump into each other and spread out until eventually they're at equilibrium. Stuff will still move back and forth, but the rate will be equal. And here's a little video. Oh, I don't know if it's going to capture this.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So diffusion, if you think about like an open space, it's a lot easier to consider. But yeah, usually in a cell, there's a separation between the inside and the outside of the cell by the plasma membrane. And so if there's a lot more of that particular substance or molecule on one side, then the concentration is a lot higher compared to the other side. So there's a gradient. A gradient means like a difference. You might have seen there's like a photo filter called a gradient, where it can have, one part of the picture darker and then turns to lighter, or like an ombre hair dye. So concentration gradient means like the difference, the change in that concentration. So substances will diffuse down their concentration gradient. They're going to want to, quote unquote, move from an area of higher concentration to an area of lower concentration because they want to spread out. So substances move down their concentration gradient, the region along which the density of a chemical substance increases or decreases. So each substance moves down its own concentration gradient, unaffected by the concentrations of other substances. So I will show a photo on the next slide that hopefully helps, or a figure. So here we have again orange dye molecules on the left and purple dye molecules on the right of the membrane. And so each dye is going to want to move its own direction. It's not going to care about, but there's purples on the other side. No, the oranges just care like there's way more of us on this side. Let's bump out and spread out so that we have more space. Kind of, So the orange molecules are going to move to the right until they're at equilibrium on either side. And then, the same for the purple dyes. So this purple dye on the right, it doesn't really care. Oh, there's a bunch more oranges on the other side. The purple's just like, hey, we're more concentrated on this side. Let's spread out until we're also on the left and we're equally spaced out from each other, purple dye molecules. So eventually each thing moves down its own concentration gradient regardless of the other substances. So the orange is going to get at equilibrium, and then the purples are going to be at equilibrium, regardless of each other. Which makes sense because in a cell there's going to be a lot of different molecules trying to get in or out. So the cell, each molecule is only moving across its own concentration gradient. It doesn't care about the other stuff. Now, a specific case of diffusion is osmosis. You might have heard about it before. Osmosis is specifically the diffusion of water molecules across a selectively permeable membrane. So it's kind of just focusing on the water molecules and their movement in or out of a membrane. So when people say, learning by osmosis, false, super false. No, osmosis is actually only water molecules, not knowledge. You can't just change definitions like that. If you were to do that, you would say it's diffusion of knowledge, not osmosis. So anyway, osmosis is very specifically diffusion of water molecules across a selectively permeable membrane. So water molecules also diffuse across the membrane from the region of higher water concentration, which is actually lower solute concentration. And then it's going to move to the area with lower water concentration and higher solute concentration. And so water will keep moving until the solute concentration is equal on both sides. So usually this happens like if there's a membrane and the membrane is permeable to water, but it's not permeable to the dye molecules, or it's not permeable to whatever that other kind of solute is. So it's like the water has to move to try to balance things out. So here's an example. Say we have a tube and there's a membrane across the middle. The membrane is permeable to water molecules, so water molecules can cross, but these are sugar molecules that are pretty big, or it's just not compatible, it can't cross. So these little green spheres are sugar molecules on either side, but there's a little bit more on the right side. And so in this case, there's On the left side, a higher concentration of water, right? There's more water per solute. And then on the other side, there's a lower concentration of water relative to the solute. And since the sugar can't cross the membrane, the water moves down its concentration gradient. So it will move from the left to the right side because it's the only thing that can cross the membrane. And so water will move to the right side. And you see this by having like that volume change. So now there's less on that side of the U and then more water on this side of the U because the water literally had to cross. Yeah, so again, that's an example of a membrane that is selectively permeable. Only some stuff can cross and not other stuff. So water crossing in this case and moving down its own concentration gradient from left to right. And that is osmosis. And so we can describe different kinds of solutions depending on how much it's going to make a cell gain or lose water. So that is what we call tonicity. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. Tonicity depends on the concentration of the solutes in the solution that can't cross the membrane relative to the concentration of the solute inside the cell. So again, I'll show a figure on the next slide. Pictures. So if the solution, again, solutes dissolved in something else, it's called a solution. If the solution has a higher concentration of solutes than the inside of the cell, then water is going to kind of want to balance out. It's going to go from an area of higher concentration of water from inside the cell to outside the cell. So if the solution has a higher concentration of these solutes than the inside, water will leave the cell, and then vice versa. If the solution has a lower concentration of solutes than the inside of the cell, water will actually enter the cell. And so there are different terms. There are 3 different terms. A solution is isotonic if the solute concentration is the same on the outside versus the inside of the cell. So here is a figure on the right. Here's a red blood cell on the top. Red blood cells normally look like this. They have a nice not spherical shape, but like a disc. Circular, but a little bit flattened. And then a little bit squished in the middle. So it's like a disc, almost like a frisbee, kind of, but not really. Anyway, so if this solution that this red blood cell is in has the same concentration of whatever solute on the outside and on the inside, then that's called isotonic. And water might enter and leave the cell, but it's going to be at like a normal rate. Because again, molecules just bump into each other and do whatever randomly. So water might enter the cell, but it's going to leave at like the same rate. So there's no net change. It's not like a lot more water is entering the cell or a lot more water is leaving the cell. It's kind of the same. So water diffuses across the membrane at the same rate in both directions. There's no net movement of water across the membrane. In a cell without a cell wall, like a red blood cell, there's no shape change. Everything is fine. It doesn't need to have a cell wall. It's fine. So the volume of a cell without a cell wall is going to be kind of stable. It's not going to like increase. It's not going to get more filled or less filled because there's going to be no change. And then in the bottom is a picture of an animal cell. A plant cell. A plant cell that's kind of shaped like a coffin that has a cell wall. And so at this point, it's not really relevant yet, but the cell is pretty happy, has its normal shape. Again, water might be leaving, but it's are entering and leaving at the same rate, so there's no change. Okay, so that is compared to a solution that's hypertonic. A solution is hypertonic if the solute concentration on the outside of the cell is greater than that inside of the cell. So say you drop a red blood cell in like really salty water. So the salt is the substance we're going to focus on, the solute that we're going to focus on. So if you drop a cell in super salty water, there might be some salt inside of a cell, but if there's a lot more salt on the outside of the cell, then that means that solute concentration is a lot higher outside the cell than inside. And water is going to move down its own concentration gradient from the higher concentration of water before to where it's a lower concentration of water because there's so much salt in the water. So then the water will leave the cell. And now this cell is all shriveled. So a net diffusion of water from inside the cell to the outside of the cell will end up causing a shape change. Cells without cell walls will lose water, shrivel, and likely die eventually in a hypertonic solution. So if you were to leave these red blood cells in a super salty solution, they just end up dying. They would lose water, shrivel, and then maybe just die off. On the bottom is a plant cell, and now you can kind of see the difference. So the cell wall maintained its shape. This cell wall still looks like a coffin. And then the inside of the cell kind of shrunk back, and so that's what we call plasmolized. Overall, the cell kind of still keeps its shape because it has the cell wall. It's not all shrinking, it's just kind of pulling back from the cell wall. And again, that's from water leaving the cell, trying to go down its concentration gradient. And then on the opposite end, it's like a Three Little Bears type situation. A solution is hypotonic if the solute concentration is less than that inside the cell. So say you drop some red blood cells in pure water. It's deionized, there's no ions or minerals or anything, in just pure water. The cells are going to be like, what the heck? There's going to be a lot less solute outside compared to inside the cell, because inside of the cell there might still be, like salt or other things, glucose maybe. But if it's in pure water, then that means the higher concentration of water is outside of the cell, and water is going to want to come in down its concentration gradient. And so in this case, the net diffusion of water is from the solution to the inside of the cell. And cells without cell walls will gain water, swell, and they might eventually burst or lyse. So just like how hydrolyse is to break something, when a cell is lysed, it's just burst, like popped. And so this cell will lyse in a hypotonic solution and burst because it doesn't have anything to keep it together. It's like so full, it just bursts. The plasma membrane can only do so much. And then at the bottom, cells with cell walls, luckily, still kind of maintain their shape. They're a little bit more swollen, but not drastically, because the cell wall is still helping them maintain shape. And so I know the terms seem super similar, but again, you can try to draw on the prefixes, like parts of the word, to help you remember which word is which, because hypotonic and hypertonic sound similar. It can be easy to confuse them at the beginning, but think of the word beginning, the prefix. Hypo. If you think of like hypoglycemia, that's when somebody has low blood sugar, for example, someone with diabetes, low blood sugar. Hypoglycemia means less than. So hypo means less than And so the concentration of the solute, the solution, is less than what's inside of the cell. So the terms always describe the solution, and it's comparing it to the inside of the cell. So hypotonic concentration is less than inside of the cell. And then hypertonic, hyper means more than. So hyperactivity is like you have a lot of energy and you have more energy than normal or than anybody else around you. So hypertonic means that compared to inside of the cell, more than. Hyper, more than. The solute concentration is greater than the inside of the cell. So that should hopefully help you remember which one was which. And I recommend drawing these. So draw, you can draw 2 cells for each of these terms. So you can draw the isotonic. You don't have to necessarily draw 2, but say there's a normal red blood cell. And then if the red blood cell stays in this, it's going to stay a nice purple, a nice pretty shape. So you don't necessarily have to draw 2. But for these, imagine, they all start out looking like this one. So you can draw it starting out looking like that. And then if you were to leave that red blood cell in this solution, what would it look like? This is what would happen. It would end up shrinking in a hypertonic solution. So I would practice drawing it out for each term. So draw a nice red blood cell, and then how it would change in a hypertonic solution, and then a normal red blood cell before, and then after you put it in a hypotonic solution. That way you have that frame of reference. Yes, that's my study tip for these. And so this is all of them together. So normally in an isotonic solution, cells will look like they normally do because they normally exist in these kinds of conditions. So a red blood cell in normal blood is going to look like this because that's its normal. It's isotonic. And then if these kinds of cells get put into a different solution, this is how it would look compared to the normal. So some environments can cause problems for cells without cell walls because they can't tolerate too much water loss or uptake, like we saw with the red blood cell. It'll either shrink or burst or lyse. So organisms that live in such environments like that need a method of osmoregulation. So osmo, like water, and regulation is trying to regulate solute concentration and water balance. So, for example, paramecium are single-celled protists, and they live in a freshwater environment, meaning that it's a very hypotonic solution. So they have to kind of figure out a way to not take up so much water that they lyse. So by living in a freshwater environment, they actually have a contractile vacuole that pumps out excess water out of the cell. So it doesn't take up too much water and then burst. And the cells of plants, some prokaryotes, fungi, and some protists also have cell walls. Although they're made of different stuff, like different kinds of molecules that make their cell walls, the concept's kind of the same. There's some kind of structure that helps protect the cells. So this is an advantage. A plant cell in a hypotonic solution will take up water and swell until the inelastic wall, so it's not going to burst, exerts back a pressure on the cell. And so then this is called turgor pressure. So at this point, the cell is called turgid or very firm, and that's actually the healthy state for most plant cells. So water your plants. If a plant cell and its surroundings are isotonic, there's no net movement of water into the cell. And plant cells will become flaccid or limp in an isotonic solution, and the plant will eventually wilt. In a hypertonic environment, plant cells will lose water. The cell shrivels and the membrane pulls away from the cell wall in multiple locations, and that's called plasmolysis. So in response, the plant will wilt and may die. So again, water your plants. And don't water them with super salty water or super sugar water. Okay, so osmosis is a case of diffusion. Again, osmosis is just focusing on the movement of water molecules. And then diffusion is just anything moving down its concentration gradient. And that's without the help of any proteins yet. So the next case of passive transport is facilitated diffusion. So in facilitated diffusion, it's diffusion that's being facilitated or helped by your protein. So transport proteins help speed up the passive movement of molecules across the plasma membrane. And transport proteins include two kinds of proteins, those channel proteins or carrier proteins. So a channel protein, has like a tunnel, and this still is just allowing diffusion to happen faster or on its own. So say this molecule is nonpolar, it can't cross on its own. It's being facilitated by a channel protein. Or there can be a carrier protein where there's a shape change and it's like just moving it across. So subheading, channel proteins, they let a specific molecule or ion cross the membrane. And there are different ones for different molecules. So aquaporins facilitate the diffusion of water specifically. There are some that are ion channels, and so they'll only let ions through, and usually only one certain ion. Remember, an ion is something that has either lost or gained an electron, and so it either has like a positive or negative charge. So ion channels let only ions through. Some ion channels, if they are called gated ion channels, there's literally like a gate. There has to be like a lock that opens up the channel, and then you could go through the tunnel. So some ion channels open or close in response to a stimulus. So there has to be something like a key that opens up this channel and then lets that kind of ion through. Maybe it's kind of like supposedly there's a Tesla tunnel or something, right? Somewhere. So it'll only let Teslas through, and there must be some kind of like check for that or something. Whatever. So in nerve cells, for example, potassium ion channels, they will open in response to an electrical stimulus. So if the cell membrane voltage gets to a certain voltage, then that triggers the channel to open, and then the potassium ions can cross. Other gated channels open in response to a chemical stimulus, so binding of a certain substance to the protein, and that is like the actual lock. So something binds, opens the gate, and then the ions can cross. And the other kind of protein that's involved in facilitated diffusion are carrier proteins. So these are the ones I was saying there's a shape change that moves the solute binding site across the membrane. This one I kind of think of as an elevator. The elevator has to come down and be on the same floor as you for it to open, let you in, and then move you to the other floor. It's not just stairs that you can go up and down on your own. So carrier proteins undergo a subtle shape change that moves the solute binding site across the membrane. And this shape change can be triggered by the binding and release of that transport molecule. So it's like it's its own lock/key. The protein will have the solute bind. When the solute binds, then that kind of triggers the shape change. Yeah. And so this is still moving stuff down its concentration gradient. So no energy inputs required. These proteins are kind of just helping it happen. But it doesn't require energy. So again, that's both facilitated diffusion. It's diffusion happening. It's facilitated by proteins, but there's no extra energy input. Compared to active transport. So the category 2 of active transport is where some of the transport proteins require energy to move substances against their concentration gradients. So it's kind of pushing molecules where they don't want to go. My favorite example of this is I have 2 sisters. One sister I'll ask to go run errands with me, and she's like, yeah, it doesn't really matter where we're going or whatever. She's just down to go with me. And then my other sister, the even younger one, she needs to be enticed. So you offer her like a Slurpee or something, or, oh, if we go here, I'll get you this. And then she'll go with you. So there's some kind of payment involved. There's energy input. You're trying to convince them and you have to give them something. And so that's kind of my comparison of passive transport and active transport. Some transport proteins require energy to move substances against their concentration gradients. She does not want to go. She doesn't want to leave the house. But if you put some energy in, she'll go with you. So active transport requires energy, usually in the form of ATP. So again, this molecule is going to come up throughout everything, the whole course, because that's the energy molecule. And so all proteins involved in active transport are carrier proteins. It's the only way you can really like force things to go where they don't want to go is by having this like shape change and move things across or against their concentration gradients. So active transport, you might think like, well, why would the cell need to do that? That sounds kind of like a waste of energy just to move things where it doesn't want to go. But sometimes it's for a purpose, or, it is for a purpose. So for example, in animal cells, the concentration of potassium ions is higher inside the cell. And the concentration of sodium ions is lower than the surroundings. And so that difference is on purpose because there's what's called a sodium-potassium pump. So it's what's maintaining that difference because it's for a reason. So transfer of a phosphate group from ATP to that sodium-potassium pump energizes the transport of potassium into the cell because you want that higher concentration of potassium inside. And then it moves and shoves sodium outside of the cell. And then that's for a reason. So here's that example of that sodium-potassium pump. So sodium is getting shoved outside the cell. You're moving it against its concentration gradient because there's less sodium inside than outside, and you're just shoveling it outside. And then the potassium in return will also get shoved inside. And so that's how it keeps that potassium high on the inside. Okay, so that is the review of passive and active transport. So there was the 3 forms of passive transport: diffusion, and osmosis falls under that, under diffusion. And then facilitated diffusion, where a protein helps diffusion happen. And then active transport, you have to invest energy, and it has to be a carrier protein that shoves things where it doesn't want to go.